Hi, Robert. Welcome to Quantum Photonics.
Hello, how are you?
Yeah, I'm good. How are you today?
Oh, I'm very good.
Yeah, so we will wait for Alexa, and
Okay.
I just pinned a link um, to your slides here at the top of the room, and
Okay.
to those who are here in the audience while we're waiting for more people to come in and another speaker to come in, please check on the link that I have pinned at the top of the room.
Looks like everything works.
Can you please check, Darcel, if the pin is the slides are working? Thank you.
Looks like everything works. Yeah.
Yeah, I agree. It does. Thank you.
Yeah, I'm able to see the slides.
I will go. I would like to welcome you and Alexa to Quantum Photonics. Thank you for accepting our invitation for you to speak today. So tonight, actually. So I would like to request Darcel, who always helps us open and close the rooms here in Quantum Photonics, to give us his music while we're pinging people in and waiting for more people to come. Darcel is a filmmaker and a musician, and
cool.
He is a writer as well. So we are honored that he helps us open and close the room as part of the team of Quantum Photonics. So in the meantime, while we wait for more people, Darcel, I would like to give the mic to you. Thank you.
Thank you, Cecile. It is an absolute pleasure to open and close these rooms. I am honored, and happy Thursday to all. And here we go. Okay, Cecile, the mic is yours. Thank you.
Thank you so much, Darcel. We are glad to have our guests here. Later on, I will try to put in the room chat the link to the laboratory of Dr. Robert Han. And please also check our Twitter account. So we have posted article that they made, that if you see a girl there in my Twitter account who is having a lovely picture with some neurons and light, that is our guest. She is our guest, Alexa. So yeah, I would like to start our the speakers today. We are very honored to have Bobby, Dr. Robert Hunt, is an associate professor of anatomy and neurobiology at the UC Irvine School of Medicine and director of the UCI Epilepsy Research Center. He obtained a Ph.D. in physiology from the University of Kentucky in 2010 under the supervision of Dr. Brett Smith, and completed a postdoctoral fellowship at the University of California, San Francisco in the laboratory of Dr. Scott Baraban. In 2015, he joined the faculty of UC Irvine, where his lab studies the principles of the nervous system development, epilepsy, and traumatic brain injury. With this content, his lab focuses on developing and applying new technologies to repair the brain. Publications from his research have appeared on the leading scientific journals like Nature Neuroscience, Neuron, Nature Communication, and Journal of Neuroscience. His research has been funded by grants from NIH, NSF, Department of Defense, and California Institute for Regenerative Medicine, including a prestigious K99 R00 Pathway to Independence Award from NINDS in 2013. Dr. Hunt has been actively involved in the epilepsy field for his entire career. He has served as a contributing editor for Epilepsy Currents since 2018. He participates actively in leadership positions within the American Epilepsy Society, and he is a member of the Scientific Advisory Board for the Lennox-Gastaut Syndrome Foundation. Yeah, our guest, also the author, Alexa Tierno, is a PhD student in the Hunt Lab at UC Irvine. She obtained a BS in neuroscience and MS in physiology at UCLA, where she worked on spinal cord injury under the supervision of Dr. Reggie Edgerton and Patricia Phelps. In her PhD studies, Alexa is creating maps of how brain circuits change after traumatic brain injury, and she is a co-first author of the paper to be discussed. So, without further ado, I welcome our guests here to Quantum Photonics, and I would like to give the mic to them.
All right. Well, hello, everyone. Thank you all for tuning in, and thank you, Cecil, for hosting us. I'm very happy to be here today and to talk with you all about my lab's research. Before we start, today is my daughter's birthday. She just turned 8 years old today. And so before we start, I want to give a shout out to Maddie Hunt. She might be listening right now. We'll be doing birthday things a little later. But anyway, what I want to talk with you all about tonight is some of the research going on in my lab right now and how it is leading to some technology that we're really excited about. What I'll do is I'm going to introduce the topic, and then I'll give you a general overview about one area of research in my lab, focus on traumatic brain injury. And then Alexa will jump in and tell you all about a project that she's been working on in the lab for her graduate studies. And at the end, we're happy to discuss anything you'd like to concerning our research, what we do here in Southern California in the lab or at UC Irvine in general. Okay, so what I want to talk to you all about today is our research on TBI, traumatic brain injury, also called TBI. You'll hear me sort of use those terms interchangeably. This is something that I've personally been working on for a long time, more than 15 years. I started in the mid-2000s, and we continue in my lab to this day working on this topic. And if you're following along in the slide deck, on slide 2 of the slide deck, I listed some facts about TBI. And when we talk about this disorder, what we're talking about is an external and mechanical force that damages the brain. And not only damages the brain, but also changes brain function. And so that's sort of the classic definition of what a traumatic brain injury is. And so things like a bump or a blow to the head would be a traumatic brain injury or a TBI. And there's many other ways that the brain can be damaged. For example, if someone has a stroke, that will also damage the brain. but a stroke wouldn't be considered a TBI because it isn't caused by an external mechanical force. So we're really talking about a blow to the head or something like that. And brain injury is actually very common. It affects a lot of people. Somewhere between 70 and 90 million people worldwide will have a TBI just this year alone, and every year. And if the injury is severe enough, it can be life-threatening. And many people will, who sustain a brain injury will die. And if the person survives, they can be at risk for a lifetime of physical or cognitive or emotional disabilities. And on this slide, I list some of those conditions that will emerge in people with TBI. Exactly what conditions emerge depends somewhat on what area of the brain was damaged, how severe it was, the biomechanics of the injury, and the type of treatment that the person receives, and so on. My lab is focused mostly on trying to understand how TBI can affect memory and the risk for epilepsy, which can be as high as 50% after a severe brain injury. Both of these are pretty major issues for people with TBI, and so this is sort of the focus of my lab. But what I wanted to do on this slide is just show all of the sort of the whole host of things that can emerge. And one of the things that my lab is trying to do here at UC Irvine is we're trying to regrow nerve cells in the brain after it's been damaged. And the reason why we're trying to do this is because there are many health conditions that involve a loss of brain cells, or a loss of their connections, or the way those connections work. And the idea is actually pretty simple. With a traumatic brain injury, or also in the case of aging or Alzheimer's disease, epilepsy, many conditions, oftentimes there are brain cells that are lost for one reason or another. And often there are no drugs that you can take that will fix the problem. Maybe you can slow it down or improve things for a little while, but the brain usually can't recover all of its normal functions, even with drugs. Even if they do work, drugs can have a lot of unwanted side effects. And so on slide 3 of the PowerPoint presentation, you can see an example of this in people with TBI. This is a chart that shows a variety of anti-seizure drugs that have been tried over the years to treat epilepsy in people with TBI. These data are now probably 15 years old or more, but the problem really hasn't changed. What this chart is showing is that none of the anti-seizure drugs that we currently have on the market, and there are about 40 that are available, none of them work to stop seizures in people with a brain injury. And so we've been trying to think about TBI a little bit differently. And what we want to know is really a couple of things. One is how are brain circuits changed by TBI? How does an injury change the structure of the brain? And also, how does it change the way the brain operates? And second, can we rewire injured brain circuits and as a therapy? And the way we're currently thinking about the brain rewiring is what we really want to do is be able to grow the missing brain cells outside of the body and then put them back in when we need to. And this type of an approach is called a cell therapy. And it's the type of thing my lab is trying to figure out how to do. If we grow the right cells and we wire them into the right areas of the brain at the right time, maybe we can fix some of these disorders that have been so difficult to treat. And so far, we've been pretty successful at doing this in different types of animals. And now we're starting to ask, is this something that we can use to help people? And so what I want to do is I want to give you an example. In the lab, we often use mice as a model system. We have machines that have been designed to give brain injuries to mice and other animals. And you can see a picture of this on slide 5. On the left is an image of the whole mouse brain. You're looking from the top of the brain. And what we do is we create a contusion injury in these animals. This is a direct blow to the head. And you can see the contusion on the right side of the brain of this mouse. And the brain of this animal is very severely damaged. And in fact, it shows up, this is several weeks after the injury, and you can see it shows up as really as a hole on the side of the brain. There's a degeneration of the tissue in the dorsal side of the brain, the cortex. And the image on the right is a slice of that brain. And you can see the damage, although it's very severe, it's mostly restricted to the area that received the blow. And so in other words, it's a pretty focal injury, but it is severe. And the neocortex is almost completely gone in the area that is damaged. And The hippocampus, which is one of the major memory centers, sits right below the neocortex. And that's also damaged, and it sort of herniates up to fill in the hole that was left by the missing cortex. And these mice, this is how we create brain injury in the lab. These mice will go on to have pretty severe memory problems, and about half of the animals will spontaneously start having seizures a few weeks after the injury. And so it's a fairly good model for these types of conditions. And one of the most striking things that we've found over the years is that after a TBI, there's a massive loss of a specific type of brain cell called an inhibitory interneuron. And these are cells that the way they work is they dampen the electrical activity in the brain. And they're also the type of brain cell that often goes missing in people and in animals with epilepsy. And you can see an example of this on slide 6. On the left, there's a picture of a mouse hippocampus. And this image was taken several weeks after a brain injury. And the white dots are all the inhibitory neurons. And they're all scattered sort of throughout the brain. And you can see them sort of scattered throughout the hippocampus here in this image. But there's far fewer of them in this brain-injured mouse compared to a mouse that didn't have a brain injury. And on the right are stains from human brains looking at the same type of inhibitory cells. And we see the same thing in human. There are fewer inhibitory neurons after a brain injury, both in mouse and human, and as well in other animals, where that have sustained an injury. And so we've sort of come up with a fairly straightforward therapy strategy, which you can see on slide 7, and it's very simple. Inhibitory interneurons are missing after a traumatic brain injury. And so what we want to know is, can we simply implant new ones into the injured area? Can we wire them in? And if we can, what would this do? And so we've been working on this. We started way back in about 2010, I think, is trying to do this in a variety of different animal models and animal conditions. On slide 8, this is an example of what this looks like in a mouse with traumatic brain injury. And what we did is we took inhibitory progenitor cells out of mouse embryos. These are embryonic cells that were destined to become inhibitory neurons, and we took them and we implanted them into the hippocampus a week after the mice had had a brain injury. And what we found is these cells migrate and move throughout the tissue, and they integrate into the brain of that host animal. And these are the green cells in the picture on the left. And you can see that there's a fairly severe injury. The neocortex is gone, and what's left is the damaged hippocampus. And these green cells are the transplanted neurons. And these new cells look and behave just like the ones that were born there naturally. You can see how well the new brain cells spread out in the damaged area, and it's really well illustrated in this image. They fill in right in the right place where native inhibitory neurons went missing, and it turns out they stay there for the life of the animal. And what we found is, at least in the mice, is that when these cells grow up in the damaged brain, they're very good at dampening the excessive electrical activity that happens after a brain injury. What they do is they increase inhibition in the brain and dampen that excessive electrical activity that often emerges after a TBI. And so these animals then go on and they will completely stop having seizures. The mice also develop memory problems after a TBI, just like many of the people do. And this gets better with the cell therapy too. And so what we found is, by implanting new inhibitory neurons into damaged part of the brain, can completely correct some of these memory problems, and we can eliminate seizures after brain injury. And this is something that drugs, all of our drugs have never been able to do. And so this, of course, is very exciting for us. What we're doing now is we're developing this technology to be used in people. Right. What I showed you is an example of where we've done sort of proof of principle studies with mouse cells into mouse brains. But if it works in a mouse, can we get this to work in a person? And so we're starting to work on those types of questions now in the lab. And so we're now growing inhibitory neurons from human pluripotent stem cells, the human version of these mouse cells that we've been working on. And these are stem cells that can turn into any cell type in the body, and we're turning them into the inhibitory neurons that go missing after TBI. And we're testing these cells in animals first. But what we're talking about is wiring new brain cells into somebody's brain. And so there are some steps that we really are working on right now to complete. We need to complete these before we could try this sort of thing in a person. So, for example, first, we really want a better idea of how inhibitory circuits are changed by TBI and how they're changed sort of across the entire brain. So far, we've spent a lot of time looking at what is happening in the damaged area for probably decades. The field has kind of focused on this damaged area of the brain, and we've learned a lot about that. But what we don't know is we don't know as much about what's happening in uninjured areas or how the damaged areas work together with the rest of the brain after an injury. And so that's something that we feel like when we're talking about wiring, brain connections and permanently changing the circuitry of the brain, we really want to understand this. Second, how exactly are these new interneurons incorporating into the injured brain circuitry? And this seems like something that we would want to know before we move forward with something that a therapy that we would try in people. And so this is where Alexa will take over, and she'll tell you about this new paper that we have from the lab in which we've started to address these types of issues. And if you're following along with the slides, Alexa will pick up on slide 10.
Okay, hi everyone, I'm Alexa, and so I'm going to be talking and going through our recently published work, where, like Bobby was saying, we really want to take like a brain-wide approach at looking to see how the brain is being rewired, not only in the injured area, but also far away from the injury site. And so to do so, we used a retrograde tracer. So what we did was we delivered a controlled cortical impact over the hippocampus, and then we wanted to trace the inhibitory interneurons, specifically somatostatin interneurons, so a subtype of interneurons, and we wanted to see what the inputs to these interneurons were and how they were being changed after traumatic brain injury. And so in order to do so, we delivered a virus that would label these somatostatin interneurons green, and then we came back 3 weeks later, delivered a rabies virus, which would then travel retrogradely, labeling the connections to these somatostatin interneurons. And then in that way, we can actually see all of the inputs to the inhibitory interneurons throughout the whole brain. And so to do so, we needed to basically have the brain intact so that we can see brain-wide what is going on after injury. And so to do so, we developed an enhanced iDisco plus brain clearing protocol, which basically would make the brain, the mouse brain, transparent. So we take out the lipids and the fats of the brain and then make the brain transparent so that we can actually see all of the connections onto these inhibitory interneurons. And so following the clearing, we label to see where these connections are using deep brain immunolabeling, and then using a light sheet microscope, we can actually image the whole brain intact, so we don't have to slice or manipulate it in any way. The connections are intact, and we can see what is going on in those connections. And then we take those images that we took, the 3D images, and we can register them to an atlas so that we can see exactly the distinct brain regions where these neurons are located, and then go in and manually count the cells within each of those brain regions. And so on slide 12 of our PowerPoint, there's a link to a video where you can actually see what the cleared brains after labeling look like, and the types of images that we're able to obtain using this new enhanced protocol. And so what we found was we mapped out all of the inputs to the somatostatin interneurons in the hippocampus. And what we found was that there was an increase in the amount of local input that these interneurons were receiving, meaning that within the hippocampus, there was more input coming into this somatostatin interneurons, so these inhibitory interneurons. As we moved further away from the hippocampus, so outside of the hippocampus, long-range input was actually decreased in the brain-injured animals. And that was evident, but when we looked at the input outside of the hippocampus, and likewise, when we looked at the contralateral side, so on the other side of the brain, we saw significant decrease of input coming onto these somatostatin interneurons. So we concluded that increase in the local input, but a decrease far away from the injury site. And so we really wanted to see what subregions of the brain are changing after injury. And so, like I said, within the hippocampus, there was an increase in the amount of input. So in slide 14, we can see areas within the hippocampus, like CA1, we saw significant increases in the number of input neurons that were synapsing onto our inhibitory interneurons. And far away from the injury site, so for example, the medial entorhinal cortex and diagonal band, which sent projections to these inhibitory interneurons, those connections were decreased in the brain-injured animals. And so in order to really see what is going on more specifically with the loss of long-range input and increase in local input, we reverse cleared the tissue. So what we did is we rehydrate the tissue, and we're able to section and immunostain as we normally do to see cell type specific changes that are going on. And so we really wanted to test out three possibilities as to why there's a loss of these long-range inputs. And so the first possibility could be that distant input neurons undergo cell death after TBI. So we tested that, and what we saw was that those neurons are not lost. So it's not that there's cell death in these regions that's causing just, the lack of input now, because those neurons are still there. So that possibility is out. The second possibility is that distant input regions are disconnected by exotomy. So what that means is that the axons are being severed, and so that's just general axon damage from the injury. That's not the case, because when we looked at the projections, they're still there, and they're still robust. So lots of projections coming into the hippocampus, so it's not just the general axon loss per se. And so what this means is that the distant input is removed in a cell-specific manner, meaning that the loss of input is cell-specific after injury. So next, looking at the local input coming into these inhibitory interneurons, again, looking at that reverse clear tissue, we wanted to see what types of cells are increasing their inputs onto the inhibitory neurons, and we saw these CA1 pyramidal cells, so these are excitatory cells, extending their processes into the region where the somatostatin interneurons are, and there was an increase in the number of inputs from these cells. And so next, we really wanted to ask, okay, so that's what's happening locally within the hippocampus, but what about far away from the injury site, an area that is not damaged directly from our TBI? And so we went over to the prefrontal cortex, and this is a region, higher order processing region, important for memory retrieval and decision making. And we chose it because it's directly contacting the hippocampus, both sending information and receiving information from the hippocampus. So we wanted to see how is this circuitry affected, even though it's not directly damaged. And so using our same viral techniques, where we would label our inhibitory interneurons in the prefrontal cortex, and then look at the inputs onto these cells, we found that similar to what we had seen at the site of injury, we see a decrease in long-range inputs, specifically from areas like the thalamus, which again is also part of this hippocampal prefrontal cortex circuit. And then when we looked at contralateral, or on the other side of the brain, those inputs onto these inhibitory neurons were also decreased. So we're seeing kind of similar types of changes that we saw within the hippocampus. And likewise, if you click on the link to the video, you can, on slide 18, you can actually see what the cleared brains look like and where all of those input neurons to the prefrontal cortex are. And so we robustly mapped out all of the input areas, both in brain-injured mice and uninjured mice, and we found 178 different regions are sending inputs to these areas. And after injury, we see changes both, like I said, on the ipsilateral side, so the side of injury, likewise on the contralateral side. So there's decreases from areas like the thalamus, which is far away, and then changes within the actual prefrontal cortex. So we see decreases from sub areas in that region. And on the contralateral side, we see a decrease, for example, in the prelimbic, which is another area of the prefrontal cortex. So again, we're seeing the same types, similar changes that we saw in the injury site, but now far away from the injury site. And this is summarized on slide 20. Again, in the hippocampus, the main takeaway from this is that there's a loss of long-range input, and it's cell type specific, and likewise an increase in local input that's also cell type specific. And then in an area that is not damaged, like the prefrontal cortex, loss of long-range input and changes in those local inputs. So our next question was, do transplanted somatostatin interneurons receive appropriate connections? So Bobby showed that our lab has transplanted interneurons, and we've been able to see, seizures decrease and memory problems improve after injury and with transplants. But how exactly are these somatostatin interneurons integrating, and are they receiving the appropriate connections? And that's what we tested next. And so, again, on slide 21, if you want to see a video of what the brain actually looks like, go ahead and click that link. But what we did was we transplanted the inhibitory interneurons into the hippocampus and let them migrate, and then we labeled them so that we can see the connections being made onto these somatostatin interneurons. And what we found was that they are indeed making local connections within the hippocampus and also outside of the hippocampus, just like the host interneurons, or the endogenous interneurons would be. So they're making the appropriate connections, like from the medial entorhinal cortex that usually sends projections into these somatostatin interneurons. Likewise, these transplanted interneurons are also receiving those same connections. And so this study really emphasized the need to look at brain-wide connections, and not just looking locally what's happening at the injury site, but the reorganization that's happening outside of the injury site. And so, next we can look at other cell types and start to map out those inputs as well, not only inputs, but also where are they projecting to and how that changes after TBI, which is an important step if we want to understand what is going on after injury in terms of rewiring those connections, especially if we want to move interneuron transplant therapy to humans. We really need to understand how the circuitry is changing overall. And so with that kind of concludes the overview of our paper. And so, I'm happy to take any questions that you may have.
Yeah, thank you, Alexa. I would just like to tell the audience to take a look at the video that they have in their presentation here on the slides. because it will help you appreciate more what they're talking about. And these are really good visuals. So if you have time, please click on the movie that they made. So yeah, I think they're ready to take questions. If we have questions to the audience, please raise your hand. If you have questions, we will bring you up here on stage. And welcome Shara, who just joined us.
Yeah, I'll probably try to take the mantle and try to ask a question. Hi, Robert, Lexa, amazing work. This is really monumental, and I think like this is probably going to be a game changer in the TBI world, especially when it comes to brain rehabilitation. That being said, I do have a couple of questions for you. For starters, I'm kind of surprised that these interneurons, or at least like the prepotent interneurons, were able to latch onto the existing network of pyramidal cells, at least like in the hippocampal or cortical area, without any kind of scaffolding, or at least without any necessity of scaffolding. I'm just wondering, is that possible for other types of populations of neurons? I guess I'm kind of curious if this could be also applied to pyramidal cells, which would be lost due to traumatic brain injury. So is that something that you're looking into? I guess I'll start with that one.
Yeah, so that's a great question. So the thing about the interneuron, so when you think about therapy, right, and you think about damage to an area of the brain, you need to, if you're going to implant new neurons and wire them into the brain, you need to cover the entire structure that needs new neurons, and so the migratory property of these cells we think is a major sort of feature of these cells that allows them to have their therapeutic properties, because we can wire in these new inhibitory neurons throughout the damaged area. It's and this has been studied for at least the last 20 or so years, the basic biology of sort of young, newly born inhibitory neurons. They're born. They're actually not born in the cortex, even though they during development, even though they end up in the cortex, they're born outside of the cortex. And during development, they'll migrate in and sort of wire in, during sort of embryonic to early postnatal development, and it's sort of this migratory sort of feature of these cells is part of their basic biology. You can pull them out, you can put them in a dish, and they'll migrate all over the dish. And what we found is we can put them in brains, and they'll migrate sort of throughout the brain. But you're exactly right. So that's, while that's a feature of inhibitory neurons, other cell types don't do that. And it would be a huge advantage, not just to get pyramidal cells to migrate, but you could think of a whole host of types of neurons throughout the brain for a variety of disorders. And the unfortunate thing is that we don't know yet what it is that gives these inhibitory cells this property to migrate, but they don't need scaffolding. They don't need the other types of things that neurons need. There have been some attempts to try to fuse inhibitory neurons to, say, an excitatory pyramidal neuron and see if that will give them the motor to migrate. We've tried some transplant sort of strategies with pyramidal neurons, both with mouse cells and with human, and they don't mind. We just can't get them to sort of move around. They're very much stuck wherever we put.
Yeah, thank you for that. I guess sort of like a tangential to this.
Yeah.
Do you actually witness this in glial cells, not just neurons, or simply inhibitory interneurons?
Oh, what do you mean? So glial cells will migrate.
Yeah. So the reason why I'm asking is that I think I came across this study. Forgot the main authors of that one, but they're actually proposing that it's probably possible to genetically target glial cells and kind of revert them back to neurons in a damaged area. So I'm asking about that possibility, like if glial cells can actually migrate, that can possibly be another alternative to your approach.
Yeah, so that's a good idea, and that's something that has been kicked around, like, I don't know, the last 10 years or so, and it is a good idea. So glial cells will migrate. They also like to proliferate after an injury. So if you get damage to some part of the brain, those cells will divide and make more of themselves. And so can you hijack the system? And also there's a developmental sort of rationale behind this as well, because neurons are born from glia, essentially. And so could we hijack that system? A number of labs have tried to do this. It's a bit controversial. So initially the studies came out saying, yes, we can say we can make these viruses that will convert a glial cell into a neuron sort of at will. And we can use that as sort of an alternative to this type of a cell therapy, like what I presented here. But lately, the last couple of years, there have been a couple of studies that have come out questioning whether or not that's actually happening, or whether those, how those viruses are working, and are you actually repopulating, the brain with new neurons, or are those viruses actually just targeting neurons, and we need to do a little bit, sort of more rigorous science to try to understand what the heck we're actually doing with these viruses. So I think it's an open question. It's a very good idea. It's a promising area. And it could be an alternative way to make some of these types of missing cells, if we can figure out how to get it to work.
Thank you. I have a few more questions, but I guess we have new people that just joined us on stage. So, Mike, over to you, Cecile.
Thank you, Abyss. I think you can continue to ask questions in a while, because I think your questions are very important. So, we have some people who joined us on stage. At the same time, there are comments on the room chat, so we will read them later. So I would like to remind everyone to give an equal opportunity for other speakers to also speak. So in the meantime, I'd like to give the mic to Vincent if you have some question or comment.
I don't know how to do this. I think I've mentioned before, I didn't suffer anything. I'm a victor, not a victim. Two men followed me home from a casino and I won a jackpot for $1,700, and they hit me with a lead pipe on the left side of my head and split open my brain, and they just stapled me shut. And soon after, I started painting like an inkjet printer, started my poetry, just increased like I was auto-writing, mathematical calculation went through the roof, spatial recognition, also testosterone reuptake, I got a lot stronger. So any points of prevalence or, helping me understand what the heck is going on in these past 3 years would be great. Thanks.
Interesting. Trying to. So it was the left side of your head, and it was.
It was on the left temporal fissure from my frontal top occipital lobe.
Interesting. And
right above.
Did you notice any vision? Anything?
What?
Any vision changes?
Maybe it got better.
Interesting. You're very lucky.
Thomas Jefferson once said, "I'm a strong believer in luck, but the harder I work, the more I receive of it." Yeah, but yes, I recognize that. Yes.
Yeah. So you're for whatever reason, you're the circuits rewired in the right way. And that's actually really interesting. It's
rare. Called the 3 R's theory that I found. It's what is it? Recruitment, rewiring, and rejuvenation.
Yeah, and you need to, that it can happen. It's rare. More often is there's damage, and it doesn't, the brain. So when the brain gets damaged. And it's not trying to make bad things happen, right? So you're trying to rewire with what you have, and sort of, it's hard for me to say, precisely what would be going on with you, because I don't really know, but like in the case, if you're missing a neuron type or a cell type, you can't replace it. You can't regrow those, but you can rewire some of the circuitry. And so the brain tries to do that. It's just usually, that's what we call maladaptive, like a maladaptive response, where the brain is rewiring, but you can kind of, it's like putting a Band-Aid on it almost. Like you can kind of get it fixed a little bit, but other things kind of emerge. But in your case, it sounds like everything was great, and you have some improved sort of functioning, and I don't know.
Gift/curses. I'll say that much. It just depends on the moment. He said that it was as if a neurosurgeon had sneezed with holding a scalpel, because it was just that precision. So they just stapled my head shut and hoped for good.
I see.
Well, I was also stabbed, like with a really big knife, the Crocodile Dundee knife, to be like certain, for my heart. Through my heart. They killed 6 people after me. Like, this wasn't some nefarious stuff. I just won some money at a casino the day before I was supposed to launch my company, which was August 29th, 2019.
Wow. You were supposed to still be here.
For some reason or another. Yeah, I guess so.
So, more people came up on stage. So, we give the mic to Mustafa. Hi, Mustafa.
Hi, Cecile. Thank you, Dr. Hans, Alexa. It was very hilarious. Your presentation really impressed me. So, congratulations.
Oh, thank you.
I'm wondering, implanting inhibitory neurons in the injured part of the brain is a very impressive idea. Once the inhibitory neurons are implanted, do you know how the brain managed to rewire them? Would the brain have kind of program for self-repair if it's helped in the right way? And does this mechanism have a relationship with the plasticity of the brain? And last question, if you don't mind, what is the role of astrocytes in the repair of damaged area? Thank you.
Okay, so your second question is very complicated. The first one I can address a little bit easier. So when we implant these new neurons in, they seem to, the brain is, and that is kind of the sort of last slide or two of what Alexa showed. And if you open up the video that we have linked with that, or open up the paper and you can read about it, those cells are wiring in the correct way. They're receiving all the right inputs. They're getting, so it's a little bit trickier than that. There's aspects of the traumatic brain injury rewiring that are still there. So after the injury, these inhibitory neurons, some of them die, some of them, the ones that survive, they are hyper innervated by the surrounding neurons, so they become these like hyper connected hubs. And there's aspects of that remain in the transplanted cells. They're pretty hyper connected as well with the sort of the local hippocampal network. But they're receiving all of the appropriate long range inputs. They're not receiving any of the inappropriate sort of connections. And so what we think is happening is that they're being wired into the brain the correct ways. in beneficial ways. Obviously, they have to be beneficial because seizures are completely eliminated in animals, and their memory issues and things like that are restored. And so that's how we think of it, is that this is we think of it as sort of an engineering issue of, you're missing your interneurons. We're wiring, enough of them back in that the brain's able to do those types of complex computations without sort of, getting too hyperexcitable and sort of going into a seizure. There's sparse coding in the hippocampus that's required for things like memory and so on. And so we think that the wiring of this is also important for that. As far as astrocytes, so astrocytes are interesting because they do a lot of things. They release a lot of sort of substances that can have effects on the neural circuitry or just on the biology of the neurons. They are, after an injury, the astrocytes are really important for walling off the injury, and they form what's called a glial scar. And what that is basically just walling off the injured area so that, you're kind of separating the healthy tissue from the damaged tissue. And at first you get this sort of, they divide and make more of themselves. And then over time you'll see this sort of tight band emerge around the injured area. And this will happen after, say, a spinal cord injury, and this will happen after traumatic brain injury. And that, in a way, is super helpful because you're kind of separating the two areas, the injured tissue. But it's also kind of a scar on the brain, and so it's hard to, if you're thinking about regeneration or rejuvenation, that wall, you can create this impenetrable wall where you can't get, anything to pass or whatnot. And then these astrocytes are really important for connecting the vasculature, and all the healthy nutrients that come out of the vasculature with the neurons and making sure that everything works properly. So they're very complex role that they're playing, and not fully understood what's going on with an injury. We know a little bit about the scarring. Recent research has shown that they're actually playing active processes in neural circuit function, but that's sort of at its infancy right now, I think, in understanding exactly what they're doing, especially after trauma.
Thank you very much.
Yeah, thank you, Mustafa. Now we go to Leanne. Hi, Leanne. Leanne.
Hi, I didn't have a question so much as just a comment regarding the previous speaker who talked about having changes in spatial or mathematical or artistic abilities, and there's many stories of that kind in the creativity literature. Many stories of people who suddenly turn into artists after they actually had brain injury and so on and so forth. I don't know the neurophysiological details of it, but it's a not unheard of phenomenon.
Yes, that's right. And yeah.
They have a name for it. It's pretty ostentatious. I don't like to use it. I just like the 3 R's theory. That feels like it encompasses it enough. Basically, I feel as though mitosis had to occur because, well, there was dead cells, so they reached out for the strongest ones, which just so happened to be on the other side of my brain, and replicated it on, the left side of my brain, making it's right above my corpus callosum, right? So it might have led to more or less connection between the two hemispheres.
I think there might be disinhibition might be playing a role as well.
One more time? I'm sorry, I'm not a physiologist. I'm just trying to do my best.
Oh, so disinhibition is where you are, so there are inhibitory neurons that inhibit signals from getting through, and if they're disinhibited, then that inhibition is stopped and they're allowed to go through.
That might explain a thing or two.
Yeah, it's a little bit more complicated than that, though, because, what you're talking about is really not just one part of the brain. You're talking about the whole thing, and it's a little bit complicated. What we're finding with the tracing studies, for example, is there's really a ripple effect that's happening across the whole brain. So damage in one part of the brain there's a rewiring, and you can think of like a massive rewiring at the injury site, but like this is rippling across the whole brain, and this is probably going to be different depending on where you get hit, what actually dies, right, and what doesn't. The dynamics of the injury, there's different types, right? So there's a contusion injury like what we're doing, which is very focal, but you can also have these diffuse injuries that are more like concussive types of injuries, and so it is very complicated.
Dr. Hunt, what could the implantation of inhibitory neuron in healthy brains yield?
What is the what?
If you implant the inhibitory neurons in healthy brain, what
happens?
Yeah, what could happen?
So they migrate, and they migrate. They will integrate just kind of the same way. We've done this sort of a number of different studies now. They will integrate the same way into the healthy brains, and you'll end up with a little bit more inhibition than you normally would. And what we found is that you can do that, and it's actually kind of remarkable because the brain doesn't necessarily need or want more inhibitory neurons, but it's taking them. But it will take them to a certain extent. So you'll get more inhibitory synapses, you'll get more inhibition, but it will plateau if you keep putting in more neurons and more neurons and more neurons. You can't. It doesn't summate. And inhibition will just plateau. And so what we found is that you can raise inhibition somewhere between 20 and 30%, and after that it will just stop. Somewhat remarkably, we've tested the animals on all kinds of different behavior tasks when we do this, and it never seems to change the animal's behavior. And we're not super certain why, because you might expect that if you over inhibit sort of something like, say, the hippocampus, which is really critical for memory, that you would end up with memory problems by tipping the scales towards too much inhibition, but that doesn't seem to be happening. On the other hand, that's great for sort of safety of this approach. What it means is that we can, implant these cells into the brain, but there's a limit to the amount of inhibition, and there's a limit, to what can happen. And so there's not really the danger that these cells would cause, major pathological sort of issues or major complications.
Yes, so thank you. Now we go to Jake. Jake?
Hi, thanks. I had a few questions. One of them, the first question is on the proliferation of these inhibitory neurons. Could it be that they're sort of seeking out areas of the brain that have a high level of activity, and so they're just naturally space themselves out that way?
I'm trying to think of the right way to answer this. So the activity, I think, comes in more into play with the sort of the wiring them in, and we haven't done a great job of evaluating this, but we may be able to, they may be sort of, well, we kind of actually do know this from some of the circuit mapping data. They're receiving a lot more connections after a brain injury. So, in the brain injured animals, there's sort of hyperactive circuits, and so the wiring is more robust than you would see in a control brain. But they're not spreading out. They're not migrating out because of activity, because you can take these cells, you can put them in a dish, and they'll do the same thing. They kind of spread out. What these cells are doing is they're actually, what I think they're doing is they're recreating what happens in normal brain development. So normally these cells are born in what we call the basal forebrain, in a specific part of the basal forebrain called the medial ganglionic eminence, and it produces most of the inner neurons in the cortex. And these cells will migrate tangentially up into the cortex, which that just means it's migrating against sort of the radioglial scaffolding that gets set up in the embryonic cortex, and then they migrate that one way up into the cortex, and they kind of come in these migratory streams. And then what they do is they kind of just spread out. Once they get to the destination in the cortex where they need to go, they kind of just spread out. And when we transplant the cells, they do exactly the same thing. And you can't see that from any of the pictures, but you can see it in our studies. When we transplant these cells into, say, the hippocampus, what they're doing is they're migrating along axon pathways in sort of different directions away from the injection site, and they migrate in these streams. And that's very similar to what they're doing in the embryo, when they migrate up into the cortex. And then after about a week, what they do is they then sort of migrate radially. They migrate and they spread out and they fill up the hippocampus. And so what I think is going on, I think there's some natural programming in these cells, and that they're born, to migrate. Mouse cells are born to migrate a certain amount, and it's about the same type of timing that you would see naturally in the embryo. They're recreating that in a transplant sort of condition, and that's what they're doing. Then at that point, sort of the circuit construction that goes on after they migrate to the right places, and how they form synapses, and the quality of those synapses, and what's going on there, I think could be activity dependent. Probably is.
Thanks. And do we understand why these inhibitory neurons do not regenerate on their own? Neurons typically don't regenerate. There's some controversial sort of takes on this, but neurons normally will not proliferate after an injury. There's a couple places in the brain that the hippocampus being one of them, that will regenerate, will just sort of throughout life regenerate neurons, but it just doesn't happen. They die, and then they don't divide. They're postmitotic, which means they're after sort of they've exited the cell cycle, and they don't return to make more of themselves. The glia will, the astrocytes, microglia, things like that. Glial cells will, but those aren't neurons. Neurons almost never regenerate. And final question, how do you get these viruses to seek out specific types of neurons and label them?
Oh, based on their genetics. So, different cell types will have different genes that they express. There's thousands of genes, and so based on what their gene expression profile is, we can target. We can do something very broad and say, if I want to target GABAergic neurons, I could target the GAD1 gene, which makes GABA, or something much more specific than that, like a transcription factor that's involved in sort of specifying their fate. But it's all driven by their gene expression.
Fascinating. Thank you.
Yeah, thank you, Jake. Now we go to Dr. Shah.
Thank you so much, Cecile, and thank you for sharing your research with us. So my question is about the potential activation of the dentate granule cells in relation with the actually preventing the seizure, because you just mentioned about that, and how it can be possible also by considering the transplanted neuron derived from the stem cells. If you can further explanation around that, that would be wonderful.
Can you say the question again? I'm having a hard time sort of understanding exactly what you want to know.
Sure. So there was a potential, you just mentioned as part of the research about the activation of the dentate granule cells. Also, they can prevent seizure. So I was just wondering if you have further information around that you can share with us.
So activation meaning the proliferation of granule cells?
Yes.
Like they divide and make more of themselves? Yeah, that happened. Yeah, so and this is not just injury, right? With epilepsy or with a lot of sort of disorders of brain or diseases or anything that sort of increases activity in the hippocampus will cause granule cells, at least in rodents, and some other lower animals will proliferate, and so you'll make more granule cells. Granule cells are funny because granule cells, if you excite them too much, they can actually start producing GABA, and they'll co-release GABA and glutamate, and so they'll be both excitatory and inhibitory under certain conditions. But they're an excitatory neuron. It's a little bit unclear, what exactly like newly generated granule neurons, what their role is going to be in epilepsy, or in some of these sort of conditions. With damage, the granule cells will start to sprout connections, and their abnormal connections, they'll kind of form these excitatory loops among themselves. And so that's been one hypothesis, is that the increase in adult neurogenesis is actually bad, not good, because those are injured cells that are now integrating into the circuitry, and they are forming these abnormal excitatory loops, and that can cause a seizure. There's other thoughts that the main output of the dentate gyrus is actually inhibition, because all these granule cells really they have these weird synapses where they really are very potent in driving interneurons, and just one granule cell can activate a whole bunch of interneurons, and so that would sort of push inhibition through the circuit, and so that maybe that would be beneficial. And then it's controversial whether or not this is a rodent phenomenon and not something that may not even be happening in humans. There's been research that's come out the last couple of years challenging the idea. It seems like it comes out every so often, challenging the idea that humans even have adult neurogenesis, and that they'll be able to make new granule neurons throughout life. And I think that's still kind of an open question that the field's kind of fighting about right now at the moment. And so what isn't happening when we do these transplants, the transplants love to go to the dentate, and one reason might be because they have the trophic factors there for making new neurons, because the dentate is always making new neurons, and so they do love to go to the dentate, but that's not the only place they go. They migrate throughout the hippocampus. We can put them in the cortex, and they migrate all over the cortex. We've put them in the amygdala, and they migrate all over the amygdala, and those areas are not neurogenic regions. And so I don't think it's really they're seeking out adult-born granule cells. They're certainly not only innervating the granule cells, and they're not only receiving inputs, although they do receive a lot of inputs from granule cells. The mapping studies have actually showed 13 or so brain regions that these cells are receiving input from, and so they're really being wired in to the entire brain. The dentate gyrus being part of it. I don't know if that answers your question, but that's kind of my thoughts on this.
Sure, thank you so much.
Yeah, thank you. I would just like to ask Alexa, because it's her paper, which we also discussed here. So this was a study based on mice. And are you having the next step to try to look at this study extended to humans?
Hi, yeah, thank you for the question. I mean, currently, like Bobby was saying, we first need to understand what is going on with transplanting interneurons derived from human pluripotent stem cells. And so before we move to a interneuron therapy for humans, we really need to understand how these cell types are integrating into the circuit and whether they are able to integrate, because perhaps there are differences between mouse and human cells, of course. So I think the next step would be to try to attempt to grow these human GABA neurons or inhibitory interneurons and transplant them and see if we see the same results that we see with the mouse transplants, doing mapping studies and looking more functionally and behaviorally at what is going on before we can move to a clinically relevant therapy.
Thank you, Alexa. I find it really fascinating what you're doing. So we will be rooting for the future studies that you will have in the lab of Bob. So we have here Abyss, I think, who still has questions. But in the meantime, while we're in the middle of this talk that you have, I would just like those on stage to flash their mic for the name of your daughter is Maddie, Bob?
Yes.
Yeah. Can we say happy birthday to Maddie, who is listening right now, the daughter of Bob? She's celebrating her 8th birthday. So happy birthday, Maddie. So let's flash our mic for her.
We need Darcel to sing us a song for happy birthday to Madiba.
Maybe later before we close the room. I think it's a very auspicious day for Bob to also be here with us. So while I had finished asking some question, maybe I'll have some questions later. I would like to give the mic to Abyss, who said that he had further questions earlier, and I will read the chat also on the room chat. Abyss.
I guess you kind of went into the kind of the question that I was about to ask. I was directed to Alexa. I think in slide 11, you're talking about the different ways of doing the imaging and sort of the labeling process. So one thing that I see in a clinical study, especially when it comes to diagnosing TBIs, is using diffuser tensor imaging that requires mapping out the projections, so it's entirely the connections. So I guess my question is, since your method is actually more targeted towards diagnosing or at least quantifying the amount of GABAergic interneurons that you have, how do you translate that to something like a clinical study or that uses diffuser tensor imaging, like I said, which is far more granular than your approach? If you're actually looking to that, I'll be very interested to know.
Yeah, that's actually a really good question. You get some good questions. DTI, there's MRI studies, and we've actually been looking at that literature, both in human and you can actually do rodent MRI. In fact, there's a lab down the hall that does this sort of all the time. They do MRI, and they actually do it on brain injury and rodents with brain injury. And there's a couple of things. So there's differences, right? So there's differences, and when you see things like DTI or whatnot, you're exactly right. You're not getting the resolution on what's going on at a circuit level. You're kind of getting this broad overview. And what's actually happening if you go to cellular resolution, like what we are doing, things can be very different in one cell type than what's happening in another cell type. And then what you're going to pick up with something like a DTI or any of these sort of brain imaging, it's going to be sort of dominated by the excitatory circuits. Because most of the brain, 80% of your neurons are excitatory and only 20% are inhibitory, even though those inhibitory neurons are really, really powerful. And so we're approaching it a little bit different from like a human imaging sort of an approach, because the way I've been having a hard time thinking about this. So, we've been thinking about it a long time. We don't know. It's like trying to drive, to Los Angeles, and they've changed the roads, but you don't have a map, and you don't know how to get there, but you have a car. And how do we understand anything about traffic, until we make a map? And so that's kind of what the approach that Alexa has been sort of doing, if that makes sense, is creating this roadmap. And then what I think we need to do is once we've figured out some of the major pathways and some of the major cell types, that are, then we need to start comparing that to what we do know in these neuroimaging studies, because we've actually come up with a couple of at least one hypothesis that seems to contradict some of these human imaging studies. So for example, there's this concept that will emerge. I'm trying to think of like the best way to say it. That the brain sort of moves away from what's called small worldness, which is that these you have different discrete brain areas, right? And they all talk to each other. And then after a brain injury, activity that flows through the network is more diffuse. There's a more diffuse activation of say the brain after a brain injury, which means that the brain areas are kind of all or none, kind of going active or not active, and we're actually finding the opposite at a cellular level for inhibitory neurons. Now, we don't know what's going on with the excitatory circuits, and we don't know what's going on with all of the brain areas, but for inhibitory neurons, we're actually seeing the opposite thing. We're seeing a hyperactivation locally, and then severing the inputs that are coming in from all the other parts of the brain. And this is happening not just in the injured area, but in the damaged area. And so this would be the opposite sort of hypothesis, that the brain is actually becoming more small world after injury, not less. And so we don't. It's a thing that I'm thinking about a lot, and it's a great question. And I don't have a full understanding of how to answer it. What we need to do is we need to do more mapping. More cell types and more brain regions. And so one of the things that we want to do is we're really trying to map the entire brain. If we can do this in a road, then we can understand, okay, this is the roadmap, this is what's happening, at least for this type of an injury. And then we want to start talking about, like, okay, how does this roadmap inform some of the things that we're seeing on function, which would be more of like what you're talking about, if that makes sense.
It does, and it's really fascinating. I didn't know about that world theory that those part of the injured part of the brain actually goes into this inhibition stage, which is really fascinating. I guess I probably would like to look into that. The other question that I had is that when you're dealing with the dentate gyrus, there are progenitor stem cells already that exist in the dentate, if I'm not mistaken. I'm really wondering. I'm not a neurobiologist here. My major is actually in kind of like a brain-computer interface.
Oh, cool.
Curious to see if how do you fight, like I said, there are progenitor stem cells that are ready to express, or at least to differentiate those stem cells in the dentate gyrus to specific population of neurons. So when you're infusing your iPSCs, I guess, like you use induced pluripotent stem cells to express GABAergic interneurons, like how do you try to maximize the expression of those inhibitory neurons, as opposed to like what key proteins exist in the dentate gyrus system, which is like, I guess, to express more pyramidal cells, if I'm not
glutamatergic cells, or I get that.
Right.
So the question is when we're trying to make inhibitory neurons, how are we able to like only make inhibitory neurons?
Yeah, that's right.
Is that yeah.
It's poorly worded.
Yeah, so that's, which is a really good question, because this therapy, I think, is going to depend on that. So with the mouse studies, we've taken mouse cells and put them into mouse brain, and those cells. So when a neuron is born, it exits the cell cycle, which means it no longer divides anymore. Once it does that, it's pretty hardwired into what it's going to become. There can be changes, but it pretty much knows what it's going to be when it grows up. And so we're taking cells that are at that stage. They have just exited cell cycle, and they are destined to become GABAergic inhibitory neurons. And we're collecting those, and then we're putting those into the brain. And we're really good at doing that with mouse cells. When when it comes to making the human version of these cells, it's a lot more tricky. So we're starting with these pluripotent stem cells, and those cells can become anything in the brain, and including brain cancer, right? And so if we transplant, actively dividing cells and we put them into your brain, you will have brain cancer, and so that obviously we don't want to do that. And we have to send those stem cells down, the path of turning into cortical neurons, and not just cortical neurons, but cortical inhibitory neurons. And then we have to collect those cells at that defined sort of stage in their development, collect them up, and then inject them into the mouse brain. And so that's kind of how we do it right now. We're doing it with viruses, where we label the cells at the exact age that we want to take them, and then we get rid of everything else. And so we know, okay, we're only putting in these inhibitory neurons. But as this emerges, depending on how easy, you can think of, the FDA approving something like that, it would be much more, of an advantage to just coax these cells to get there and make those cells on their own and not make anything else. And so that's going to be, one of the hurdles that we have to sort of solve in moving this to a human therapy. But there are others, actually, because the human neurons actually take a long time to develop. You think of gestation in a human is much longer in a mouse than it is in a mouse. And so, we see that replicated, in these human neurons as well. So it takes them a long time to sort of grow and mature. And so we don't fully understand how that will translate to wiring cells into a circuit if they have to sit there, in the brain, for 6, 7, 8 months, and integrate. And what we can, we're just starting those studies now. And so there's a lot of open questions like that, that we're approaching. But the short answer is that, we can make them into inhibitory neurons by taking them sort of at the right age, when they've, they know what they want to be when they grow up. They just haven't grown up. So they're kind of like newborn neurons.
Yeah, thank you for that. And I guess like one final thing, like based on what you're discussing about that, like how, like time that, the gestation time that human neurons actually take in order to probably approve this for, or at least they go for the FDA route. I'm curious if you actually looked into using organoids as opposed to like, directly implanting this to non-human primates or for that matter. If you consider that to be like a stepping stone to see if your infusion technique actually.
Yeah. So you could. I think how you make the cells isn't as important as what you make, right? And so brain organoids, there's all kinds of different types. They're kind of cool, right? Because it's a miniature brain that you can kind of watch and develop, and you can fuse them together, and you can make different types and do a lot of neat tricks, in vitro. A few people have transplanted them. A few groups have transplanted organoids, but they haven't tried to make necessarily specific types of organoids. They just, we made brain organoids and we put them into a mouse brain. Those organoids grow. When you do that, you put an organoid, into a mouse brain, it will grow, and it will be a literal cell replacement. I mean, it will start to kill off the mouse brain, and then it will be a big organoid in there. And they'll send out, projections and things like that, and connect with the mouse brain just the same as any cell transplant. But I don't think that's a good way to think about therapy. The biggest challenge is, what do you make? Because you could make an organoid, that's similar to, you could envision creating a brain organoid that makes the inhibitory, like an inhibitory organoid. And then you take those cells and you kind of dissociate them and use them for therapy. I could see that working. The challenge has been in the field has been to make bona fide inhibitory neurons. There's a lot of work, preliminary work that is sort of lay the groundwork for making inhibitory neurons. There's a lot of people that will say, hey, I made an inhibitory neuron, but But a bona fide inhibitory neuron, and there's different types. There's a conversation sort of earlier about disinhibition versus inhibition, because some interneurons would be bad for transplant purposes because they're going to contact other inhibitory neurons and turn those cells off, and so they would disinhibit the brain. We want to inhibit the brain. And then also there's all kinds of programming issues that go into this, because you're kind of coaxing neurons. Sometimes they'll express things that they kind of look like interneurons, but they're not actually. They don't behave the same way because they've been sort of generated from this stem cell, and so they might stain for a marker and not for another marker, or they, when we do electrical recordings from them, they don't have the signature that you would expect from an inhibitory neuron. So there's a lot of things that go into this. I think you could use a brain organoid. I don't think it matters, though. I think what matters is the type of cell that you create, and that these cells go into the brain, that they migrate throughout the injured part of the brain, and that they integrate in very predictable ways. That's what we want to do.
Gotcha. Thank you.
Those are great questions.
Yeah. So, Bobby, you're here on stage. Would you like to comment or ask a question, Bobby?
Yes, please. Can you hear me?
Yep.
Okay?
Yep.
A quick clarifying question before a second more kind of broader question. You mentioned you leverage viruses to kind of kill off the cells that you don't want to transplant in this process. You actually leverage viruses to kill off like what you don't need.
We're actually doing the opposite. So we're using viruses to label the ones that we want. Because what we can do is we can label them and fluoresce them a color, and then we can collect them.
Nice. I think I followed like similar paradigms. Awesome. And then thank you for that information. And just a more broader question: is there like a canonical like a common understanding that inhibitory neurons are they like intertwined with memory? Like memory in humans, is there kind of like research there, or not so much?
That's a really good question, and I don't know how to answer that quickly. So yes and no. I think the field is getting there. So the work we've been doing is really pushing us towards what is the role of inhibition in interneurons in memory. Clearly, when you're missing interneurons, so what you need for memory, and this is a really complicated sort of question. So yes and no. So you need all the neuron types for memory, really. But what the interneurons are doing is they are dampening sort of the circuit, and what they're allowing is for a sparse coding, a sparse activation of neurons as signal travels through, say, the hippocampus. And that's really, really important because if all of your neurons sort of activate at the same time and then turn off at the same time, that's sort of the definition of a seizure. They're all synchronously activating and deactivating at a time. You're not going to have a memory. You're not going to be able to have things like differentiate between different contexts, for example. And so we think that this sparse coding is really, really important for memory. A lot of that's being controlled by interneurons. And so we're very interested in this area. I think sort of the neuroscience field in general is kind of moving in that area, in that direction, although that's not the only direction. For years and years, there's been sort of different theories and hypotheses that have kind of emerged over the years, and from how memories are stored and how memories are sort of very complicated. I don't know if I could answer that sort of right now on the stage, but if that makes sense, I think what the interneurons are doing, they're really important for the sparse coding, and I think that's playing a direct role in memory.
Awesome. I'll definitely look into this further, especially sparse coding. As you were talking, I was just thinking of it. I actually was looking up like the definitions of excitatory and inhibitory neurons.
Uh-huh.
And when I read about excitatory neurons, I was thinking, how would I have memory if I just had a constant stream of just like information? Yep. Versus like maybe the inhibitory neurons can create some sort of like cycles, like from a graph theory kind of thing or something. I don't know. Like maybe the inhibitory neurons create like pockets of memory. But definitely, thank you, and I'll look forward to learning more about sparse coding. Thank you.
I have a question.
Yeah.
When you say inhibitory, could you elaborate just a little bit on that? What is being inhibited?
Okay, Alexa, you should try this one. You're very quiet over there, and then I'll step in.
Yeah, absolutely. So what we mean by inhibitory is that the neuron is sending an inhibitory signal to another neuron. So basically, it releases a neurotransmitter that binds, which is just like a molecule. It binds to another neuron and thereby decreases the activity of that, what we call postsynaptic neuron. And that's what we mean by inhibitory, thereby inhibiting the postsynaptic cell.
So maybe like a risk you might be more, less adverse to.
Well, so here's a good analogy. You can think of your car. You have a gas pedal and you have a brake. The excitatory neurons are like the gas pedal. They push and they make the car go forward. And the inhibitory neurons are like the brake. They stop things, and in the most basic sort of sense. And so if you think about driving down the highway and you don't have your brake, you can't stop. You're going to go fast, and you're not going to be able to stop, and you're going to crash. And that kind of is what, in a very, very crude sense, is what's happening if you kill all of your inhibitory neurons in your brain. There's this hyperexcitability because all you have left are excitatory neurons. They're activating, the brain. It's basically an activation or a deactivation of the cells. Does that make sense?
So much sense.
Okay, good.
I feel like a child, like a 3-year-old. I'm so excitable about the smallest things.
Oh no, that's great. I'm glad I could explain that.
Yeah, no, thank you so much. I really, really appreciate it. And please don't hold back, in fear of hurting my feelings or anything like that, because I'm here as a scientist just as everyone else is. I went from marketing to now I'm a integrated ML AI practitioner and engineer, as well as, I'm a big fan of sales rooms.
Oh, cool. Yeah. This is fun. Yeah, there's a lot of computer people in the room too.
That's the other side of things, but that afterwards.
Please, I have 2 questions.
Yeah.
Thank you. Why do you choose inhibitory neuron? Are there other possibilities or other choices?
So okay. So yes and no. So one of the most, it's the most basic, simple. When I think of questions and when I think of how to answer questions, I think of what is the most simple thing I can do, and I'll do that experiment. The reason to choose inhibitory neurons is the tight link between inhibition and epilepsy, the loss of inhibition in epilepsy. So for the last almost 100 years, we've known that if you lose inhibition, you will the brain becomes more at risk for a seizure and for developing epilepsy. And this has been shown sort of a million different ways. One of the oldest sort of thoughts that I can think of right now off the top of my head is back in the 1940s, clinicians would actually put a drug on the brain that would block inhibition, and they would use that drug to reveal the seizure areas of the brain. Then they would go in and they would cut that part out because surgery is one of the solutions for, say, an epileptic disorder. And what would happen is when you block the inhibition, the brain would seize. And they didn't even really fully understand what they were doing. It was just sort of a technique to identify those epileptic circuits. If I block inhibition in an animal, that animal will have a seizure. And then in the brain injured animals and in people with brain injury, they're also missing these inhibitory neurons. And so it was the simplest sort of question that we could ask. Like, since this is the case, what would happen if we put the inner neurons back in?
Yes, it makes sense. But it's specific to epileptic disease.
So it's not. We think, yeah, it's not specific to epilepsy. So I mean, we've been working with memory and brain injury, which is this sort of brain injury project was born out of some of my research, almost 10 years ago, looking at this same type of thing for epilepsy. But you see the same thing in Alzheimer's. You see the loss of neurons. There's a compromised inhibition, and there are labs looking at manipulating inhibition in Alzheimer's disease. And so anywhere where you have, I think, the loss of interneurons, the decrease in inhibition, and high-level circuits, this type of an approach may work, but needs to be kind of worked out for each one of these disorders individually.
Thank you. I presume that this type of experiments will open very promising perspectives for research in general, and more particularly for brain surgery and reconstructive surgery. I would like to come back to Cecile's question. When it comes to the human brain, what are the limits of transposing experimental results from animals to human? And what are the ethical precautions to observe in this type of experiments when it's comes to human?
Yeah, that's a very tricky question. I think if you do it depends on what type of experiment you do. I think if you do a bad experiment in an animal, I think your chances of translating to human are poor. If you do a good experiment, I think it's good. I think there's been a lot of talk about translation in animal research and how well it translates, and it's tricky because we all do experiments, and some experiments are better than others, and sometimes when something fails, you go back and you look and say, oh yeah, that should have failed because this was not a very we missed this, or we missed that, or we weren't careful here or there. With the case of interneuron transplantation, I think it's really interesting. So there's other sort of experiments and projects and results that have happened sort of along the way, especially in the context of epilepsy. And I think that this is very translatable, and will eventually be part of the medicine that we have for people with epilepsy and neurotrauma. So as an example, my old group, the group that I used to work with, when I came down here to start my lab at UC Irvine, they actually started growing cells from pigs, and just a couple of years ago, transplanted those cells into sea lions with epilepsy that had washed on shore, and were able to cure a sea lion with the same exact approach that we presented today. And so, that's a great proof of principle in terms of like, would this work in a person? Well, it worked in a mouse and it worked in a sea lion. Why wouldn't this approach work, for a person, at least with epilepsy? Other disorders and other diseases, I think it's very context dependent. What might translate and what not. Certainly, drug studies, you can kind of think the same way. For a long time, a lot of the drugs that were developed in mice specifically for traumatic brain injury, the focus was always on sparing the whole. We want to make the whole smaller. We want to spare tissue. And I don't think that's going to be the right approach. All of those drugs that have moved on to clinical trial have failed, and I think they should fail because I think that's the wrong question. We don't necessarily want to spare tissue and keep that tissue alive, in a brain if that tissue is going to be bad and cause bad things to happen. We might be happy with that tissue dying and then, coming in and wiring the circuitry, in a good way. And so, I think it's a tricky question. It's a good one, and something that I think we need to talk a lot about. But I guess I can stop there.
Thank you, Alexa.
Yeah, so we have Michael who came on stage. Michael, would you like to ask a question or have a comment?
Cecile, if you don't mind, I have to go. I have, but before I want to thank Dr. Hunt and Alexa, not only for coming here to share with us their article, but especially for their research. And I wish you full success. Thank you very much.
Well, thank you. It's been a pleasure to be here.
Thank you.
Hello, this is Michael. Am I on?
Yep.
Well, I also echo the gratitude for this. There's so many keywords and topics to Google going forward. I have a personal interest in CNS injury, having been rendered paraplegic 40 odd years ago. TBI, it wasn't even on the radar. But I'll throw out some things, see if any of them pique your interest. I'm thinking about the directionality of signals and the distribution of these inhibitory cells. I wonder if there's a guidepost in the Human Connectome project. Are there any of those random pieces relate to what you're doing? If not,
in a sense, a lot of the connectome work right now is trying to understand what the connections are in the brain, especially the human brain. Animals as well. There's a lot of different projects that are just trying to purely map just what the normal connections are. Presumably the next step would then be to start looking at, like, okay, how does it change under different contexts? One of them could be injury, but it's a massive undertaking.
Yes.
Just to kind of understand, because there's so many, there's billions of neurons, and those are billions of connections, and each cell type wires itself in a different way.
I watched as colleagues like Fred Gage move from tissue studies down here near San Diego to now at the SOC and using genomics. So I'm just following the broad outlines as a layperson interested in. The studies of the connectome. I guess those are embryological, adult, all different stages. There's so much.
Exactly. Yeah, it's a huge undertaking.
But we made headway with the what I thought was intimidating, the genomic progress. But now you're working with those tools.
Yeah.
I'll leave it at that. I really wanted to thank you and jump in to express my appreciation.
Awesome. Thank you.
So I'm done. Thank you.
Yeah, thank you, Michael. So, I would just like to mention that Bobby and Alexa have been here for about 1 hour and almost 2 hours now, and I just don't know how much time do they have for us. In the meantime, I saw Eric and 2 Erics, actually, I.O. and another Eric, who are up on stage. So I'd like to ask you, Bobby and Alexa.
So I'm happy to take a couple more questions, and then I probably should run.
Thank you so much. So Eric Ochilowski, I almost admit you up here because of your...
Cool. Thank you. So my question is, in terms of the sequence and coding of the, memories and other things, can you comment on where that sequence information is stored or how it is stored? Because I know it's connections of networks, and then we see with neural networks that they kind of correspond to certain functions and groups and so on, so they're able to resolve many transformations. But in terms of, where is that song stored, and how is it that they're able to reconstruct that? I know there's some mention of sparsely distributed memory, but that seems to be more of an artifact that we've exploited in artificial neural networks. But if you could comment on that, I'd appreciate it. Thanks.
Yeah, that's a tough question. I can try. Initially, a lot of memory is stored in hippocampus, and you can... We know this because if you take it out, and then you aren't able to form new memories. But it's not permanently stored there, because if you take it out, you can remember past events. You just can't remember recent events. It sort of depends on where you are and what type of memory you're talking about, whether that's a context that you're trying to like a contextual memory, or a spatial navigation, or things like that. There's a couple of ideas. One idea has been that they're, it's actually kind of popular. This idea of engrams, that when you have an experience, your neurons will become activated in a certain collection of them. And there's been a number of really interesting rodent studies that have shown that you can actually tag those neurons when they're activated, during an experience. Say, for example, like a fearful experience. And then you can go in and you can reactivate them at a later time point, and that animal will become afraid, even though it's in a different and non-fearful context. And so that's led to this idea that you could have like this population of different combinations of neurons are representing sort of the memory. And so if you activate the right combination, you would get, say, one memory versus another. I don't think that that's going to play out to be the full story, although it's sort of a neat experimental trick, because you think of from birth until death, we all, of an entire human, you would very quickly run out of combinations of neurons, at least in sort of the more major memory centers of the brain. And so that seems unlikely. Also, we also know, the storage of memory will move from, say, the hippocampus and is thought to be dispersed sort of more broadly throughout the neocortex, and how that is happening is not really fully known.
So do you conceive of any way of, let's say, pinging that kind of experience for the rodent without having to have that original waveform or activity signature of the neurons? Do you imagine there might be a future where we could construct, let's say, I don't know, some sort of pinging mechanism based on some sort of like GPT-3 does, hey, language, and then it does some interpolation. Yeah. We could ask some sort of question and say, hey, is there any connection or collection of neurons that have this sort of activity? Or because that seems like an exhaustive thing to run through. That seems almost like you would have to run through combinations on a safe. So other than pinging a bunch of safe combinations, I don't know how you would engage that highly amazing fluid interpolation engine that we call, human understanding or the neocortex to some extent.
Yeah, I think I think everything's possible. I'm always sort of a positive believer in that we can always do things, but I think you're right. I think there's a problem. It's a challenge. It's how do you do it? I don't know. We're not there yet. That's kind of the reason why we've kind of gone to some of these mapping sort of studies in the first place, is we need to understand, sort of the roadmap that we're working with. If we're talking about, something like a memory or just even an experience, activating collections of neurons or not, or whether it's in the connections themselves, the synapses or whatnot, we need to know what all of it looks like first. And I think this has been the idea behind sort of this broad neuroscience push to map the entire brain, because we need to know sort of the material that we're working with. And then after that, there's biology that needs to be done. We need to understand how does this work, because we don't know a lot of the things I think that you're talking about, but I think that we can know. We're getting really good now at different types of sensors and being able to see what's going on in the different cells, and I think that the imaging technology is just going to explode over the next decade on being able to image with cameras outside of the brain, outside of the body, and image what's going on at a cellular resolution inside the brain, when different things are happening. And I think that's probably the only way to really understand it, because that's the only way. You need to see the entire thing happen sort of in real time.
That's awesome. Now you're talking my language, because I work on such sensors, so that's
cool.
A great way to pass on the question for the next person. Thanks.
Yeah, thank you, Eric. So, we also have another Eric here on stage. So, yeah, Eric, I give the mic to you.
That's very kind of you. Thank you very much, Steve, for having me up on here. Very interesting. Voltage first, second. The question I have might appear to go off topic a bit, but for me, it will answer quite a lot of questions about recovery within the human brain as it is. My interesting question, I'll ask you, really, obviously the interesting talk, you probably know the answer to this, that's why I've jumped up. I think it may be quite relevant to the exact topic, so I apologise for in advance for that. But the question is, when the memory is basically the signals are going backwards and forwards in the brain, when you see something, when the light changes in your eye, you hear a different sound, that sends a signal through your brain continuously, which then sparks a whole series of events in your brain backwards and forwards. But obviously brain waves going backwards and forwards, when those signals sort of arguably stop, if they actually technically ever do, what is actually causing those brain waves to continue going backwards and forwards, if I may?
Okay, so let me see if I can kind of understand this. So the brain has sort of this natural oscillation that never goes away. It'll change, if you're sleeping or if you're awake. There's and we call them rhythms or oscillations, and they happen at different frequencies. They never go away. They never change. They do change. They never go away. For example, alpha waves in the frontal cortex are related, when those are sort of increased and high, that's linked. We know that that's linked with, say, things like attention. I have your attention, for example. And what neurons are doing, on top of those waves, that's happening. That's the electrical field sort of throughout the brain, and in different brain regions, you have different sort of oscillatory activities, and those are crashing into each other all the time. At the same time, if you go down a level and you look at what's an individual neuron doing, one nerve cell, they'll be firing action potentials, and they'll be turned on or off, throughout that. And so if I see something and light comes into my eye, in the back of my head, right in the back, in the occipital lobe, signal will pick up and it will fire, depending on sort of the tuning, of those neurons to different images and light and things like that. But all of that's happening on top of these sort of broader scale waves. And some neurons are more likely to be active at different peaks and troughs of individual frequencies. So it gets it starts to get complex, but it never turns off.
So those brainwaves essentially sort of an element of randomness being sort of stimulated.
Oh, sometimes, yeah. Sometimes there is noise. There's noise in the system, and that's
a coordinated noise then.
Yeah, and it's supposed to be, in one way you could think of that as being a good thing, right? Because if you want to pull out, so you're tasked, if you're a neuron or you're a brain structure, you're tasked with trying to figure out, there's information coming all the time. There's action potentials, which are the neurons coming on and off, but there's also synaptic connections where just little sort of amounts of transmitter are crossing synapses, and within all of that, you have to figure out which signal is important and which is not. And so this is kind of the challenge of neurophysiology, is to figure this out, and we don't fully know it and understand what's going on here, but all of this is happening at the same time, and within all of it, it's making up our thoughts and our sort of how our brain is responding to experience in the world.
That's cool. I was wondering, dare I ask, do you think that the brain waves themselves are essentially the sole contributing factor to consciousness?
Oh, I have no idea. I have no idea. I know when you're. But not to consciousness. You can. you can have, there's shifts in, brain rhythms, but you still have them, if you're unconscious. If you're dead, you don't have them. So death certainly will stop all brain activity. But until then, you do have, but relating what's happening at the rhythm level, because that's kind of a bird's eye view of what the brain's doing. It's looking at the sort of the field potential activity and relating to what's happening at a neuron level and a synapse level and how that even relates to a thought. We're not there yet.
Yeah. Well, I wonder whether those brain waves essentially trigger random thoughts. That's why you sit there doing nothing, and all of a sudden something comes back to you from a while ago. Whether it's actually that sort of randomness that actually causes you to think about stuff and causes thoughts and process to happen that you wouldn't otherwise be there. And that ultimately is the consciousness.
I think that's there. I think those thoughts though are also triggered, right? By you don't always spontaneously think of, say, what I had for lunch yesterday, but if I smell something, it triggers, oh yeah, I had a sandwich for lunch yesterday, and so there's triggers on top of that as well. It's very complex. Yeah.
That's great. Much appreciated. Thank you so much.
Anyway, I think I probably need to run. It's 7:00 in my time.
Bobby and Alexa. I would just like to mention to Eric here. We had the previous room with the lab of Dimitris Pinoutsis from MIT and I think City University. Please check that out. He was talking about electric fields. He was likening electric fields as conductors of the neurons. And also, meaning to say maybe they might not be constant. They could be just moving and there could be waves, but there is something that is always trying to hold it in place, the control, and to keep the harmony within the band. So it's here on the replay. Please check that out.
Definitely. That's amazing. I'll have to definitely check that out. And this is just my apologies. One day that I try to be productive, I miss an epic talk. So way to go for me. I'm glad there's a replay though. Thanks.
Yeah. Thank you so much, Bobby and Alexa for answering all kinds of questions here. And I hope we can invite you again. And we will prepare to wrap up the room. Do you have any last comment, Bob and Alexa?
Just thank you all for coming and talking. This was great. It was a lot of fun. A lot of you brought up, some great questions about our research, and that was fun, and really some conversation about things that we don't regularly talk about necessarily in the lab, but are fun to talk about and kick around even if we don't know the answers. And so thank you all for coming. This was, I thought, a lot of fun.
Yeah, absolutely. Thank you for having us here and letting us talk about our research, and hopefully we can come back and present some of our more exciting work in the future.
Yeah, we also would like to inform you that there is a replay of this room which will be available in your profile maybe on or before half day, and you can share it, copy and share it. So I would just like to announce our next room. I'm not yet sure, but maybe tomorrow we will have AI, neuroscience, and physics news. So please check that out. That's going to be tomorrow evening. I will decide maybe in an hour or so. And then in the morning, we will have a room about decentralization of data design. So we have a researcher from Imperial College who's going to talk to us about this. So please follow Quantum Photonics, and thank you so much to our guests. We have, I don't know if Abyss wants to have some final words. I'm bringing Darcel to have a countdown here. So Abyss.
Anna, I would like to thank Alexa for their time and for sharing their very interesting work, and I'm really kind of hopeful and that this is going to have really positive effects in the future. So thank you for your time and for sharing your research with us.
Thank you. So one last comment from me. Just happy birthday, Maddie, And I now would like Bob and Alexa to experience how we close the rooms here in quantum photonics. Yeah, I give the mic to Darcel.
Thank you, Cecile, and I would like to echo a biz. Thank you, Alexa and Hunt. It was very interesting, and as always, I learned a lot. So to close the room, we're going to start our countdown with lucky number 7. Is everybody ready?
Ready.
7.
7.
Route 66.
6.
Abyss 5.
5.
Go. Cuatro.
4.
Eric Trace.
3.
Dos Seques.
2.
Alexa and Hunt, you are both number 1. Go ahead and call out our last number.
1.
Yes.
Yes, so thank you, Bob and Alexa. Yeah, see you again here in Quantum Photonics. Yeah, bye for now. Have a fun weekend. So, yeah, it's to everyone. See you in the next rooms. Good night.
Bye, everyone. Good night. Have good dreams, everyone.